All right, so this is the last section in Chapter 5, and we're going to be talking about genomics and bioinformatics. So, through our advancements in technology, the Sanger sequencer, different things like that, we can actually sequence entire genomes. So we can take the DNA out of an organism and sequence the entire thing. And then with all of that information, we can study it, we can look for different organisms that have mutations in certain genes, we can compare these genes with other organisms, so on and so forth. So there's a lot that you can do just simply by knowing an organism's genome. So genomics is just simply the study of genomes. So it can include the sequencing of those genomes, it can include comparisons of the genomes within a certain species or across different species, because oftentimes you'll find similar genes in different species. Okay? It also works to discover the function of these genes, and that's where the really difficult part comes in. Okay, because generally, in order to figure out what that gene does, you have to find somebody who has a mutation in that gene. Okay, so the gene is not functional or it's functioning different, and then you can start to see what that gene actually does. Okay, so gene sequencing really is the whole thing that genomics is based on. So this is determining the order of DNA nucleotides in genes or in genomes. We're going to see in a couple chapters in the next semester why it's so important to know the order of these nucleotides. Okay, so if you know anything about coding, you've got all those zeros and ones, and they have to be in a specific order in order to get the code to work the way you want it to. And DNA is really the same kind of thing, except instead of just zeros and ones, you have 4 different options, A, C, T, or G. So the genomes of several different organisms have been completely sequenced. So these are a couple examples of the ones that we have. Okay, so E. coli is the simplest out of all of these. It only has 4.6 million base pairs, or bases. Okay, so it's relatively small. It is also used a lot, or very, very frequently in research labs. So it's really nice that we have the full genome. It's been studied quite a bit, so we really know kind of what we're doing with E. coli. Fruit flies, they are my nemesis. I hate fruit flies, but they are also used pretty frequently in different research labs. I had a professor in college who was obsessed with them. It was very strange. I mean, and that's me saying that, so yeah, it was really weird. Yeast you have at 12.1 million bases. So yeast, also a really good organism for research. And then you've got a couple different species there. And you see humans in the middle there. So this is one thing that scientists were relatively surprised about when we first started sequencing these genomes, is that humans don't have the largest genome. Okay, they thought that the larger the genome, the more complex and intelligent the species would be. But you see vanilla right there, which nobody would say that vanilla is smarter than humans or more complex than humans or anything like that. Vanilla has quite a bit bigger genome size than humans do, over double of what humans do. So it doesn't really equate the size of the genome to the complexity of the species or anything like that. So all of this was started by what's known as the Human Genome Project. So this started in the early 90s, I believe it was, and it was basically a race to see what technique, what group could sequence the entire human genome first. So, yeah, basically it was a contest between scientists across the world. And there were a couple different techniques they were looking at on what was the best way to sequence these DNA base pairs and the human chromosomes. And so the scientists could basically pick whichever method they thought was the best and help out with that effort. But yeah, I know it sounds really weird, but science competition, it's pretty cool. So, the Human Genome Project analyzed DNA from just a couple donors, right? And scientists are still working to identify and map human genes. It's a lot more complex than it might sound, simply because there is a lot of information included in those 3,000 million or 3 billion base pairs. Okay, so actually I wanted to show you what the Human Genome Project looks like as of now. Okay, so this is done through a website called NCBI. So it's the National Center for Biotechnological Information. So NCBI has the entire human genome on it. And when you go to their website, it looks like this. So this is showing you all 23 chromosomes, okay? And then you've got a couple different references down here for taking it to from DNA to proteins and things like that. Not going to get too far into that. But I wanted to show you what the genome looks like. So we'll look at chromosome 7 here. If you know the specific location of a gene, you can search for it here. Otherwise, because we're just kind of looking at it, this is what it looks like when you get into it. So this is relatively zoomed out, and it shows you that we're looking at chromosome 7 right in this region. You can zoom out from here. You can also zoom in. And then you've got a couple different mutations are annotated here. So with this genome, they put in the entire genome, and then scientists all around the world will come in and say, okay, I found a patient that has a mutation in this region. This is the genetic code of that mutation, and then these are the symptoms, the diseases that this person has. Okay, and then other scientists will come in and they'll see that and they'll think, okay, so I can develop a research question on this and try to prove how exactly that mutation gives rise to whatever disease they have. So you can see here, this one has an epidermal growth factor receptor mutation. Okay, so epidermal growth, that would be skin growth. Okay, so there is something wrong with how the skin cells grow in this. If you want, let's see, where is the zoom? We can actually zoom into the sequence. Okay, so you can see the DNA sequence here. You can see mutations, okay, the variants is what they call them on here. And, you know, there's a lot of information that goes into all of this. And the thing that you have to think is, this is not a quick kind of thing. This has been going on since the 90s, where they have been working to compile all of this information. And it really is across the entire world. It's scientists all over the world looking into all of this and trying to basically decode the human genome. So it's a huge process. And obviously there are a couple different things that you can look at here. There are also extra tabs for looking at specifically cancer-causing genes or genes that are related to a specific disease, different things like that. So there is a lot that goes into all of this. And you can also kind of move around on the chromosome. It's a little slow, but you have to think about all of the information that it's looking at. You can also widen your view. Okay, so we're looking at a lot here. And you can see the different mutations, all of these different mutations in this area. Okay, so this is what the human genome looks like at this point. Okay, so there is quite a bit of information that you can find on here. If you ever get bored one day, I don't know, I think this is really cool to look at. But yeah, it's up to you if you want to start looking through all of this. That's great. Anyway, yeah, so you can also switch between your different chromosomes. So we're on chromosome 1 now, and it tells you up here what location you're in. Homo sapiens just tells you you're looking at the human genome, so on and so forth. And so you can see all of the different things. And generally they will group different ones, different similar mutations together. So anyway, I think it's really pretty cool, but I mean, that's me. So moving right along. Okay, so all of this technology allows scientists to study and compare genes and proteins. So we didn't really look at the protein side of NCBI. That's where it gets super complicated, because it's not just a simple match the gene with the protein. There's a lot of modifications that happen in between, so it can be kind of difficult to kind of figure out what protein comes from what gene. So there's a lot of work being done there as well. And all of this gave rise, all of this technology gave rise to a new field of study called bioinformatics. So this is the use of computer databases to organize and analyze biological data, because you have terabytes of information with the human genome, and then with all of the analysis that scientists have been doing on it and the annotations and everything, there's even more data. So have to be able to use some kind of computer program in order to analyze any of that. There are certain indicators of a location of a gene in the DNA, so they will actually use computer programs to scan through the DNA and highlight areas that they think there's a gene there, so humans don't have to try to look through all of this information and go crazy just staring at A, T, G's, and C's. So you also have what's known as a DNA microarray. So these are used to study the expression or going from DNA to proteins of many genes at once. They end up looking kind of like this, where they will light up different colors based on positive, negative results. So these are relatively nice because you do get to look at hundreds to thousands of results all at once. And oftentimes you can get the computer to analyze a lot of this as well. So that's pretty nice. And then you also have proteomics, which is the study and comparison of proteins on their own. So bioinformatics, using computer databases to organize and analyze this data. And then you have proteomics, which is comparisons of proteins specifically. Okay, so we've talked a little bit about gene therapy back at the beginning of the semester when I gave you that video on CRISPR. So CRISPR is being used a lot in gene therapy now, or it's starting into the process, but genetics provides a basis for new medical treatments. So if it's a genetic disease, then oftentimes it's like in the past, it's kind of just been, well, it's genetic, we can't really do anything. You don't have any kind of treatable disease or anything like that. So it's just trying to deal with the symptoms, not trying to treat anything. But with the advent of genetic screening and genetic testing, therapy, all of this, we are actually seeing more and more options available for this. And we're actually going to have to pause here. I didn't realize how close to the end of this video we were, so we'll pick up here on the next video.